Hi everyone, welcome to our discussion on safety in the clinical laboratory. Before we proceed with the different activities that we have prepared for this sem, let's talk about first safety inside the lab. It's because as medical technologists, we are always exposed to different types of hazards. This is also to ensure not only your health and safety, but also the health and safety of your co-workers in the laboratory. These are the hazards that are found in the clinical laboratory. We have biological, sharps, chemical, radioactive hazards, electrical hazards, fire or explosive hazards, as well as physical hazards. This table summarizes the hazards I have mentioned earlier, what are their sources, and the possible injuries that one may get when exposed to these hazards. So don't worry because as we go on with our discussion, you would be able to know and understand more of these hazards so that when you encounter them in the laboratory, you would also be able to know what to do. We start with biological hazard. So this is our biohazard symbol. Biological hazards are hazards brought about by living organisms. These living organisms can infect both humans and animals. The most common source of biological hazards in the laboratory are from patient specimens, usually in the form of blood products and other body fluids like urine, cerebrospinal fluid, seminal fluid, pleural fluid, and the like. These specimens are due for serologic or immunologic testing. Occupational Safety Health Act or OSHA mandated that all laboratory workers must observe the universal precaution. We all know that the universal precaution says that all laboratory specimens must be handled as though they are potentially infectious. Even if the sample comes from a family member, it still has the capacity to infect both humans and animals. So next, we have the chain of infection. The chain of infection is composed of three main components that are linked together, and in order for a disease or infection to occur, there must be a consistent link between these three components. Now, what are the three components of the chain of infection? We have the source or the infectious agent, the susceptible host, and the mode of transmission. But there is another comprehensive illustration of the chain of infection with three additional components. These three additional components are the portal of entry, the portal of exit, and the reservoir. I have mentioned earlier that in order for an infection to occur, there must be a consistent link between these components. But our goal here is to actually break the chain of infection. And the only way to break the chain of infection is through hand washing. So hand washing, when done correctly, it's the most effective way to prevent the spread of communicable diseases in all settings. So we need to sing the happy birthday song when we perform the hand washing. You have to sing it twice while doing the hand washing activity. Now, let's refresh our mind about the steps on the hand washing activity. So here are the steps on how to perform proper hand washing. So first, we turn on the faucet and we wet our hands with warm water. Second, we apply soap, preferably an antibacterial soap. Rub using both hands, create a lather, apply friction, and loosen debris. The third step is to remove dirt in between fingers, remove dirt from fingernails, and including the wrist. So do this for 20 seconds. The fourth step is to rinse or wash the hands in a downward position, and this is done to prevent recontamination of the wrist. Fifth, get a dry paper towel and wipe both hands. The next step is to get another paper towel and use this one to turn off the faucet. And lastly, all used paper towels must be discarded in appropriate waste containers. So there you have it, the different steps on how to perform proper hand washing. Next, we go to our personal protective equipment. The personal protective equipment is also a very important factor, especially when we work inside a laboratory as medical technologists. Your personal protective equipment includes gloves, fluid-resistant gowns, goggles, masks, and face shields. Gloves should be worn when in contact with patient or when we are trying to process specimens inside the laboratory or when we are manipulating laboratory equipment. When specimens are collected, gloves must be changed between every patient. Fluid-resistant lab gowns with cuffs should be worn completely buttoned and gloves should be pulled over the cuffs. And lastly, for the mucous membranes of the eyes, nose, and mouth, which are very exposed to specimen splashes and aerosols, the medical technologists must be wearing goggles, masks, and face shield. So these are photos showing you guys the components of our personal protective equipment. So we have here the disposable latex gloves, we have the goggles, surgical masks, fluid-resistant gown, and we also have the face shield. So again, please do not try to process specimens if you are not wearing your personal protective equipment. Biological waste disposal. So all biological waste except urine must be placed in appropriate containers labeled with the biohazard symbol. We discard the urine by pouring it into a laboratory sink. Avoid splashing, then flush with water. Disinfection of the sink using a 1:5 or 1:10 dilution of sodium hypochlorite should be performed daily. Daily means we have to perform disinfection and decontamination of the working area every day. Next, we have the Sharps hazard. So when we talk about Sharps hazard, it's about broken or other sharp glass, sharp or broken plastic, razor blades or scalpels, needles, and other sharp metal. So these types of hazard or waste, they are not disposed in regular trash baskets, but they are disposed in a special container we call the Sharps container. All sharps must be disposed in labeled plastic sharps containers. So we have here no recapping of needles because needles may be recapped only by using a mechanical device. The reason for this is that when you try to recap a needle, there is always a possibility of getting accidental punctures, especially if you are not being careful. So it only makes sense that when you try to discard a sharps, it should be done by a mechanical device. These are photos showing you guys syringes with used needles, especially that as medical technologists, one of our main jobs in the laboratory is to collect blood sample from our patient. So we must handle this type of waste carefully because it can transmit infection. So not only the used needles from our syringes, but we also have broken glasswares. Okay, so we have to discard them in appropriate sharps container. Now, what are the characteristics of an ideal sharps container? It must be puncture proof, it must be properly labeled, so you should be able to see the word sharps on the container. It must be labeled with a biohazard symbol. So if you take a look at this photo of our sharps container, there is the presence of your biohazard symbol. It must have a cover to prevent spillage of contents, and ideally should not be made of fragile material. And lastly, your sharps container must contain grooves to facilitate ease of needle removal. Because after you collect the sample from your patient, since there is no recapping or recapping is prohibited, you just have to place your used needle on that groove and then twist the barrel. Automatically, the needle will fall inside the sharps container. Next, we have the chemical hazard. For the chemical hazard, most of the reagents being used inside the hematology laboratory may cause immediate harm to the lab personnel when not handled carefully. Some of these reagents are carcinogenic and pose a long-term effect or may cause an immediate effect upon contact or inhalation. That's why when we handle our chemicals inside the laboratory, it is also required that the person will wear the personal protective equipment. Aside from wearing the personal protective equipment, these reagents must be handled inside a fume hood because the fume hood provides an added protection. Okay. Chemical hazard is considered a chemical is considered hazardous if it is determined to be flammable, explosive, toxic or poisonous, corrosive, irritant, or considered as an environmental hazard. Okay. At this point, we can see here a person under the emergency shower. So, in cases wherein there are accidental chemical spills to your body or to your eyes, we have this following apparatus inside the laboratory that will help us eliminate or lessen or prevent us from being exposed to the chemical. So we have here our emergency shower. So if you take a look at this photo, the person immediately went to under the shower and pulled the lever. So a strong force of water comes out from this portion here. Okay, so you don't need to take off your clothes when you're going to use the emergency shower. For accidental spills concerning the eyes, all you have to do is go to the emergency eye wash station and then you push this one, push this portion here, and automatically a strong force of water will actually come out from this portion, and all you have to do is to position your head near this portion so that the water will reach your eyes. For chemical hazards, we also have this other symbol, so kindly take note of these symbols. This may come in handy during quiz. Okay? Then for chemical spills, the best first aid is to flush the area with large amounts of water for at least 15 minutes and then seek medical attention. So it's not the other way around wherein you go to the emergency room first before you flush the area with water. So again, it's always flush the area with water before going to see a doctor. All laboratory personnel should know the location and proper use of emergency showers and eye wash station. And lastly, do not try to neutralize chemicals that come in contact with the skin. For chemical handling, we have other precautions here that acid should always be added to water to avoid the possibility of sudden splashing caused by rapid generation of heat in some chemical reactions. So it's not the other way around wherein you have to add water to the acid. It's always acid to water. Next is pipetting by mouth is unacceptable in the laboratory. I mean, if the sample is a urine sample, would you allow yourself to pipette the sample using your mouth? So I don't think so. Okay, so nowadays, we already have serological pipettes, automatic pipettes that aid in the transfer of samples from one container to another. So you don't have any reason to use your mouth when transferring the sample to your containers. Then we have the chemical hygiene plan. So we still have OSHA. OSHA also requires all facilities that use hazardous chemicals to have written chemical hygiene plan. Okay, so what can we see in the chemical hygiene plan? We can see there appropriate work practices, standard operating procedures, proper wearing of our personal protective equipment, engineering controls such as fume hoods and flammable safety cabinet, employee training requirements, and medical consultation guidelines. For the chemical labeling, this is also a very important factor in safe handling of our chemicals, because if the medical technologists know what he or she is manipulating or holding inside the laboratory, then he or she can also practice safety in terms of dealing with the chemical. So labeling is very important. There should be a label description of that particular hazard. If the chemical is a poisonous chemical, is it a corrosive chemical, or a carcinogenic chemical? The National Fire Protection Association, or the NFPA, has developed the standard system for the identification of the fire hazard material. So this NFPA symbol is very significant because it informs firefighters of the hazards they may encounter in a particular area. So this is our NFPA symbol, or the National Fire Protection Association symbol. It's shaped like a diamond and it has 4 quadrants. And each quadrant of the NFPA symbol has a specific color. This specific color in each quadrant signifies a specific type of hazard. For example, for the red quadrant of the NFPA symbol, it symbolizes the fire or the flammability hazard. The yellow quadrant symbolizes the instability hazard. The white quadrant symbolizes the specific hazard, and the blue quadrant symbolizes the health hazard. And as you may notice, in each of the hazards seen in the National Fire Protection Association symbol, there are gradings, okay, from 0 to 4, except for specific hazard because it does not have a grading. Instead, it has a specific description about the chemical being handled. For example, the chemical is acidic, it's basic, it's corrosive, or the chemical is an oxidizer. Okay? The next slide, this is also the same as the previous slide, but this time it's not in a diamond shape symbol, but more or less, this gives you an idea that again, the NFPA symbol has 4 quadrants, and each quadrant has different colors, and these hazards have specific grading. So I want you also to familiarize the different gradings and descriptions of each quadrant of the NFPA symbol. Next, we have the MSDS, or the Material Safety Data Sheet. This one is a document that contains information on the potential hazards and how to work safely with the chemical product. So, the MSDS includes the following: the physical and chemical characteristics of a chemical, the fire and explosion potential, reactivity potential, health hazards and emergency first aid procedures, methods for safe handling and disposal, primary routes of entry, and exposure limits and carcinogenic potential. So all this information seen in your MSDS will help you handle the chemical very well, especially if there are accidents inside the laboratory due to that chemical, or when you're exposed to that certain chemical. At least you know how to handle the situation because you have a guide through the material safety data sheet. Then we have physical hazards. Okay, physical hazard is a hazard which threatens the safety of an individual through contact or non-contact with the hazardous material. So it does not mean that when there is a physical hazard, you're always dealing with chemical. It could be in any form, like mechanical, electrical, fire, or radioactive hazard. So it doesn't have to be always chemical when we talk about hazards, okay? So there are other forms of hazards inside the laboratory. Okay, for the mechanical hazard, so you have here two illustrations. So we can see that mechanical hazards are like trip hazard or tip over hazard. So do not move this equipment without mechanical assistance. So these are examples of mechanical hazard. Okay. Usually mechanical hazards result to uh, accidents inside the laboratory. That's why before you manipulate a certain equipment or move a certain apparatus inside the laboratory, please look for these signs, okay, and read these signs before you do something, before you move that apparatus or that laboratory equipment. Next, we have electrical hazard. This is also another kind of physical hazard. So due to electricity supplying equipment and other electricity-dependent instruments, remember that inside the laboratory, we have a lot of laboratory equipment such as centrifuge, centrifuge, we have refrigerators, rotators, water baths, electric stoves, projectors, biosafety cabinets, and etc. that require the supply of electricity to work. So let's just be careful when we handle these types of laboratory equipment. So what are the possible causes of electrical hazards? The first one here is overloading of electrical connection. Okay, we also have improperly labeled electrical sockets, chipped electrical wires connecting the instrument, improperly grounded electrical equipment or instrument, and damaged electrical equipment. So please be careful when handling equipments inside the lab that make use of electricity. Kindly check the wirings, the outlets, the plugs because you may not know that while you're holding the wire of that equipment and then it's already broken, you may get electrocuted. So these photos illustrate the different situations wherein inside the laboratory wherein there is source of electrical hazard. Like for this one, this is overloading of the electrical socket. This one, plugging the equipment using wet hands. And then this one, if you take a look at the wire, the wirings are already exposed, and worse, this one, the person already got electrocuted. Now, precautionary measures in handling electrical equipment. All electric equipment must be grounded with three-pronged plugs. So, if electric shock accidents occur, never touch the person or the equipment involved. You can actually help in a situation if you can notice that your classmate is already um, undergoing electric shock. You can kick your patient away from the source of the electricity. Okay? But never hold your classmate using your bare hands. Turn off the circuit breaker, unplug the equipment, move the equipment using a non-conductive glass or wood object. Avoid overloading electrical sockets with multiple connections. Okay? And then, check the wires and plug for any signs of damages before using. Avoid using wires or plugs with damages. Check for the proper voltage of the equipment and check the equipment or machines for any sign of electrical failure such as unusual smoke, unusual burning smell. So if you can notice that your machine inside the laboratory produces an unusual smoke, please avoid using that kind of machine. Then we have fire or explosion hazard. So if a fire is discovered inside the laboratory, we all know that the most common acronym to use here is RACE. So our RACE, it has a meaning. R means rescue, rescue anyone in immediate danger. Okay. A, alarm, activate the institutional fire alarm system. C, contain, close all doors to potentially affected areas. So this is also to cut the supply of oxygen to the area, because in the presence of oxygen, your fire will get bigger. Then E, extinguish, attempt to extinguish the fire and if possible exit the area. Okay, and then we also have here how to operate a fire extinguisher. We have the acronym PASS. So it also has a meaning. P means pull the pin. Then A, aim nozzle at the base of the fire, not on the top of the fire, but on the base of the fire. S for squeeze the handle, and S is to sweep the nozzle from side to side. So that's how you operate the fire extinguisher. And then we also have types of fire and fire extinguisher. So we have types A, B, C, D, and E. I want you to familiarize this table. It's very easy because for type A, these are ordinary combustibles like cloth, paper, rubbish, plastics, and woods. And then type B, we have flammable liquids. This is very easy to remember because when we say type B and when we say liquid, in vernacular, it's basa. Okay, so flammable liquids, type B, basa. And then we have type C, electrical equipment and motor switches. That's C, corrente. Type D, flammable metals. And then type E, we have detonation or arsenal fire. Then we also have type K. Type K is the kitchen fire. Okay. Next, we have radioactive hazards. This is not a very common hazard encountered inside the laboratory because we don't usually deal with radioactive chemicals except for immunoserology section, because sometimes they make use of radioactive dyes in detecting certain analyte in the sample, but not all the time. Not all laboratory are designed to deal with radioactive hazards. Okay, then we have physical hazards, general precaution. Avoid running in rooms and hallways. Watch for wet floors. Bend knees when lifting heavy objects. Keep long hair pulled back, especially for girls with long hair. You have to tie your hair. Avoid dangling jewelry. Maintain a clean, organized work area, and wear closed-toe shoes that provide maximum support and comfort. Okay. And then we have the healthcare waste disposal. For the healthcare waste disposal, um, remember that our laboratory generates different types of waste every day, and they are not all discarded in the same container, or they are not disposed in the same container. So we have to segregate the wastes that we generate, and then we discard or dispose them in appropriate waste containers. So for black containers, we have non-infectious dry waste. So what are examples of non-infectious dry waste? We have papers, plastics, like that. Then we have green colored containers for non-infectious wet waste. Example, kitchen and dietary waste. Yellow containers or bags, infectious and pathological waste. Samples are cottons with blood, or cottons used for blood collection, band-aids, gauze with visible blood. Those are examples of infectious or pathological waste. Yellow with black band, chemical waste including those with heavy metals like mercury, copper. Okay, those are examples of heavy metals. And then we have orange for radioactive waste and red for sharps and pressurized containers. So what are examples of pressurized containers? If you notice, containers for mosquito spray like Baygon, those are examples of pressurized containers. Okay, so these are the references that I use to make this PowerPoint. Okay, so that ends our lecture on laboratory safety, and I hope that you will have a fun time in hematology section. Okay, so thank you.